Okay, so today we're going to be discussing chapter 12, part 2. So the discussion of the first part has been posted to Moodle. It's also obviously like linked and available on YouTube if you're following my channel on YouTube. Again, I will pre-apologize. I get kind of excited about this stuff. I'm hoping that I can break it down and give you some kind of just tips in terms of understanding this material, just because it can get a little difficult when you're talking about things like ionic concentrations and electrical changes and kind of how all those things come together. But hopefully analogies will be helpful, and we'll also get a chance to discuss them in class as well. So, but if you do have any questions about this material, please do reach out. Okay, so where we're at and where we're going in terms of assignments, please make sure that you're staying on top of things like your study guide, that you're attending either Tuesdays or Thursdays class. Your quiz is going to be due by Friday, like normal for chapter 12. Your pro section is due by Friday at midnight. There are 3 parts to this pro section, so just make sure you complete it by Friday at midnight. And then lastly, your adaptive practice for chapter 13 is due next Monday. And that's going to be kind of a continuation on talking about the central nervous system. So this week is kind of just like a nice overview, and then we're going to get a little bit more specific in the following chapters, talking about things like cranial nerves and then regions of the brain, and really looking at some anatomical differences between regions of the brain. Okay, so we have a pretty hefty to-do list today. So we're going to talk about ion channels, which I did discuss in the last lecture. So if that is at all confusing to you, maybe watch the end of the last lecture and the beginning of this one, because I might explain the ion channels slightly differently, and maybe it'll hit with you in one way or the other. The reason I'm kind of doing the review here is because I think it's so important to understanding exactly how the electrical conductance of a neuron is established. And so we'll essentially just kind of, again, review what these ion channels are. Talk a lot about action potentials, summation of neurotransmitters and synapses. We'll talk about neurotransmitters and how they function, differences between spatial and temporal summation, and yeah, diseases, talk a little bit about neurogenesis. So it's going to be a fun lecture. Okay, so electrical signals and neurons, an overview. So, okay. So neurons, again, last time we talked about the different cell types in the nervous system. Neurons are the communicative types of cells within the nervous system. They're the ones that are sending and receiving signals. So when we talk about sending and receiving signals between neurons, it's an electrical type of, communication that's occurring. Now, when I talk about the electrical communication is basically how the electrical signal travels within the neuron itself. And so all of these inputs are reaching out into the cellular space through dendritic projections. All of that information kind of gets brought to the cell body. And then at the cell body, that cell has to decide whether or not it's going to send that signal out into the world, and it does that via an action potential. So once all of that information has kind of been summed or summated, we'll talk about that later. If it reaches a certain threshold, that cell decides, hey, I'm going to send this signal out into the world, and it's going to do so via an action potential down that axonal projection. And so if any of these words don't necessarily seem familiar to you, go ahead and go back and watch lecture 1. Did describe a lot of cellular anatomy and just some of the definitions of some of these different words I'm using. So short and long distances were graded potentials. Graded potentials are kind of sub-threshold potentials, and we'll talk about that, meaning they don't necessarily hit that little bump, that little mark in which an action potential needs to fire. So the production of an AP or GP depends on the existing resting membrane potential and the existence of certain ion channels. So if you remember last time, and I'm just going to keep saying this because, again, I think it's useful to recognize there are certain channels that are important to know, certain ions that are important to know. So sodium, potassium, chloride, and then I'll also put calcium here, but calcium's a little different in terms of its effects. It doesn't necessarily affect resting membrane potential as much, but these 3 guys right here do, essentially. And again, we'll get to be talking about the salty banana plenty of this lecture. So just recognize that those ion channels are going to be the ones that we'll be focusing on during this lecture. Okay, so this is a very dynamic process, and by dynamic, I mean there's ions moving in, there's ions moving out, there's channels opening, there's channels closing. So sometimes, my word symphony might not be enough of an explanation. So if that's the case, do feel free to watch YouTube videos about this, or this link here is going to be being providing you essentially, again, a dynamic representation of the process, which again, if you're a visual learner, sometimes using these videos probably isn't the worst thing. Okay, so kind of again, overlapping last lecture, graded potentials and action potentials. Here you can see the sensory receptor. Sensory receptors, again, this is a pressure receptor. So this pressure receptor is having a nerve action potential that's firing. You can see that because it's a black arrow. So once that black arrow hits, you've got that, you have it hitting. And then once it gets to this little neuron right here, you can see that it's changed into a graded potential, meaning it's, you know, a little bit sub-threshold, and that's being sent all the way up here to the left side of the brain. Interesting just to note that this is the right side of the body and it changes over to the left side of the brain. We'll talk about that a little bit later on in following chapters. And then here you have your cerebral cortex that is causing a response. And so you can see that there is a graded potential here. Again, the graded potential is subthreshold, but then enough of them summate to cause an action potential. This is the threshold point. So this is the threshold. So there you have your action potential, and that action potential is coming down and sending the message to move the muscle down the efferent pathway. So afferent, efferent, afferent, efferent. Okay. And that's just me being silly and again trying to remind you of things that we talked about last lecture. Okay. Again, if you are visual, use these videos. Okay, so types of ion channels. So leak channels are very important. This, I think it's just important to kind of remind yourself, ooh, that's kind of fun, that when we're talking about movement of ions, like I kind of said last time, and maybe this is not the best way to describe it, but when you're an ion, you always want to move from an area of high concentration and to an area of low concentration. So again, you can think of ions being kind of like antisocial individuals saying, hey, you know, there's too many people at this party. I need to get out of here. It is too hot. There's too many, you know, things bumping against me. So they always want to go out of the club, away from kind of where the action's happening. So they'll always go from an area of high concentration to a low area of low concentration. And so in the example that we see here, you can see that there is a high concentration of potassium inside the cell, and it's basically forcing it to an area of low concentration outside the cell. So if you have a leak channel that kind of just spontaneously opens and closes, you know, just it's like a doorway that opens and closes, and every now and again, a potassium ion sees its chance and just goes outside. And so because of that, it kind of helps the cell keep that negative internal resting membrane potential. We'll talk about that more here in a second. But again, you've got potassium that is high on the inside of the cell and wanting to go outside of the cell. Last time I kind of, again, keep talking to you about these ions. Think of the neuron as a salty banana. So the reason it's a banana is because bananas have a lot of potassium. So there's a lot of potassium inside the banana. There's a lot of salt and chloride on the outside of the banana, and that's what makes it the salty banana type of situation. Okay, the next, so that's a leak channel. Leak channel, again, it's leaky because it just kind of randomly opens and closes. Next, we have the ligand-gated ion channel, and this is often the type of ion channel that's correlated to the binding of a neurotransmitter. And so we have lots of different neurotransmitters in our brain, and we'll talk about those later today. So the example that we see here is acetylcholine, which is one type of neurotransmitter, GABA, glutamate. You've probably heard of serotonin and dopamine. Those are all neurotransmitters. So they bind to this little binding set here. It's almost like a lock and key, which people don't really like it when you oversimplify it like that, because it's far more complicated. But this key kind of unlocks this ion channel, opens it up, and those ions are able to flow through. Again, the ion is going to flow from an area of high concentration to an area of low concentration. So if this channel allows for things like sodium to pass, sodium is going to go from outside the cell to inside the cell. And if you have potassium, potassium is going to go from inside the cell to outside the cell. So you can kind of see the movement of all of these different ions. And honestly, it might even just be worth drawing an example of like each one of these channels and walking your way through them. Okay, mechanically gated ion channels. The first kind of pressure receptor that we saw there, pressure receptors are mechanically gated. So if any pressure or disruption to a mechanical surface occurs, so here you can see That's probably not a great color for that. If you see, still not a great color. If you see a disruption of the essentially surface of a plasma membrane of a cell, it's going to cause that gate to open, and because of that, then you can have influx of ions or efflux of ions outside the cell. So in this case, it would be maybe sodium or let's say chloride that want to go inside the cell, and in the case of the type of ion that might want to go outside the cell, that would probably be potassium. So it just kind of depends on what that particular channel allows to go through. So channels have very specific shapes of opening, meaning that not all ions are able to pass through all channels. So with that being said, some channels are only permeable to sodium, some channels are only permeable to chloride, some channels are only permeable to potassium. Some channels are wide enough that they allow multiple types of ions through, but for the most part, they're very specific. Again, it's kind of like a doorway that only a certain size of individual or ion can move through, and it really, in most cases, does come down to size, and in some cases, it does come down to charge in terms of the types of charge that lie inside of that ion channel, but that's getting very specific. Okay. Lastly, this one is probably one of the more important ones to talk about today because we will be talking about voltage-gated ion channels. And okay, so it's probably the most important thing to point out here is that generally the cells sit at a pretty negative place, right around -70. It varies a little bit between cell types, but for neurons, it's right around -70. Okay, so you can see this little gate here is closed. You can see you've got lots of potassium inside the cell, and again, it wants to go out. Now, let's say that that voltage increases to -50. At -50, that voltage-gated channel opens, and you've got potassium flowing out. So if we were to draw that on a graph, and this being voltage, and your x-axis being time, let's say over time you see an increase in voltage, and then once this is -70, -70, let's say once you've hit -50, what happens is you're going to have that voltage-gated channel open and a huge increase in the voltage or decrease in the voltage, kind of depending on what happens. And in this case, because you have potassium leaving, this would cause a decrease in the voltage because potassium is leaving. But that's not. But we'll talk about again kind of how all of these ions are triggered, these different channels to open at different times, and kind of the function that they have in cell signaling. So voltage-gated ion channels get triggered at a very specific voltage. I'll talk about how exactly it happens here in this next couple slides. Okay, this is a wonderful table over, outlining leak channels, ligand-gated channels, mechanically gated channels, and voltage-gated channels. It tells you where they're located. I went over this last chapter. If you have any questions about this, I'm happy to answer it. We've got a lot to cover today, so I'm not going to read the table right now. Okay, resting membrane potential, also sometimes referred to as RMP. You can kind of see where that comes from. So resting membrane potential, also called RMP. And this is essentially the voltage at which cells naturally just chill out at. So like I said before, neurons usually hang out at around -70 millivolts. Millivolts is kind of the units. Voltage, again, is what we're measuring because we're measuring electrical changes. When we say -70 millivolts, this is the charge inside the cell. So if we look at the neuron, it's sitting at -70. There's its axon. And when I say -70, just recognize that it's basically the difference between the inside of the cell. That's what this little triangle is saying, that, okay, you've gained access to the inside of the cell. So you're measuring the difference between the inside of the cell and the outside of the cell. So as comparatively you measure those, you're sitting at -70. So outside of the cell essentially is at 0, and inside the cell is at -70. So I guess it's worth just pointing that out because this -70 is, I guess, relative to what the external ionic concentration is. So you're kind of measuring, it's a nice measurement of the differences in the charges of the external and internal of ion concentrations. That's what generates the voltage. Hopefully that doesn't confuse you too much, but if you have any questions, again, we'll discuss it to extreme lengths on Thursday. Again, if you're a very visual learner, here is a video. Okay, so RMP as defined by the text is the membrane of a non-conducting neuron is positive outside and negative inside. So kind of what I was just saying about the relative measurement inside and outside. This is determined by unequal distribution of ions across the plasma membrane. So we've talked about this pretty extensively, right? You've got a bunch of sodium outside the cell, sodium, 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 and some chloride, some chloride, some chloride. And we'll just say you've got just a little bit of sodium on the inside and a little bit of chloride on the inside. And then you've got lots of potassium on the inside. And then maybe just a little bit of potassium on the outside. Okay, so unequal distribution of ions across the plasma membrane and the selective permeability of the neuron's membrane to both sodium and potassium. So sodium can come in and potassium can leave. There are a couple different ion channels that allow that. We'll talk about those. Most anions cannot leave the cell. So again, cation is positive, anion is negative, and the anion, for the most part, again, that we speak of most times is chloride. And then sodium potassium pumps. So sodium potassium pumps are little pumps in your body that basically work pretty continuously to establish this difference in distribution that we see. So you might ask yourself, why is there so much sodium outside the cell and why is there so much potassium inside the cell? And sodium potassium pumps achieve that. Sodium potassium pumps are constantly sending sodium outside the cell and constantly pumping potassium in. This pump, we'll just actually draw the pump here. It looks just kind of like two little, this is the channel. So it's always sending sodium out, always potassium in. What's interesting about this is that it also requires ATP. If you remember from like chapter 1, essentially, ATP is the energy molecule of the cell. So your brain requires a ton of energy because it's using up so much ATP to establish this concentration gradient that you see here. Like it is unreal how much glucose your brain uses and essentially use it or metabolizes to generate ATP for the sodium potassium channel or the sodium potassium pump to essentially maintain this gradient of lots of potassium or lots of potassium inside the cell and lots of sodium outside the cell. So it's always doing this. And there's a ratio here of this, but I'm not going to get too much more in the weeds there because we're not going to talk about that extensively, but I know I don't want to overwhelm you, but I do want to make sure that this is clear. Okay, so going into some figures that are provided by the text. So outside of my drawings, so like I said, you've got your plasma membrane here, and the concentration of positive and negative ions on each side. So you've got your positive ion, positive ion, positive ion, negative ion, negative ion, negative ion. So you can see kind of relative numbers, equal number of positive and negative charges in most of the cytosol. So this is a case, electrical potential difference across the plasma membrane, equal numbers. Okay, so this is kind of, again, starting if you had a zero resting membrane potential, if you had equal numbers of positive and negatives on each side of the plasma membrane. Okay, now here you can see, like I was drawing my own little version of this. If you put a little electrode inside the cell and you measure it kind of outside the cell, the overall kind of difference that you see is roughly -70. So you've got more of a positive charge on the outside of the cell relative to this more negative charge on the inside of the cell. So again, it's just kind of worth recognizing here the differences between the positive and negative charges. And essentially, it's just a relative term. So let's say the ionic concentration exchange outside the cell, that's going to affect your resting membrane potential. If it changes inside the cell, again, that's going to affect your resting membrane potential. And so when action potentials occur, again, this is going to be the result of positive ions flowing into the cell and the cell becoming more positive. And that's, again, kind of alluding to the next couple slides. Okay, so factors that contribute to resting membrane potential. So we've talked about some of these previously, right? You have potassium leak channels. So again, potassium is kind of like one of those ions that's like, hey, there's just too many people at this party. I would like to go from an area of high concentration to an area of low concentration. So if that's possible, I'd like to do that, thanks. So because of potassium's kind of antisocial behavior, it's gonna go from inside to the outside if it gets a chance to. So a leak channel just randomly opens and closes and potassium makes its way out. You have a sodium link channel, does kind of the same thing, randomly opens and closes, and potassium sodium, because it is higher concentration outside of the cell, comes into the cell. But this does not kind of overpower the potassium link channel. It doesn't do this enough to make the inside of the cell positive. Okay, so you've got these link channels with kind of exchanging both potassium and sodium. So you can see potassium's going out, sodium's coming in, potassium's going out. And then as I kind of was explaining the sodium potassium channel or pump, in that case, you have the cell using energy to essentially move sodium outside the cell and potassium inside of the cell. So if we remember again, chapters 1 or 2, whichever chapter it was, when we were talking about active transport is active because it requires energy. Energy is required because what you're doing is you're forcing these ions against their concentration gradient, meaning that you're taking it from an area of low concentration and making it go to an area of high concentration. That's the case for sodium. And then in the case of potassium, you're doing the exact same thing where you're going from an area of low concentration outside the cell to an area of high concentration. And so again, you're moving these two ions, what we call against their concentration gradient, against where they want to be. So it's almost like you have that friend that, well, I keep saying antisocial, but you just have this friend who isn't into crowds, but you have to physically shove them back into the club saying, hey buddy, we're having a great time. Go back into the club. So that requires your energy to move him back into the club. And the cell essentially does the same thing. It's like, okay, you go back into the club, go back into the club, and that's what's going to help us keep this cell at a negative charge. So it requires energy to do so. Does anybody even really go to a club? Not in Missoula, I don't think, but I'm sure there are people who've lived in cities who do. Okay, other things to kind of point out here, other things that contribute to resting membrane potential is the fact that the cell is just filled with proteins as well. So these little red balls here are showing you proteins, and proteins for the most part carry a negative charge, depending on the protein, but a lot of them do carry a net negative charge. So that also contributes to that resting membrane potential. So the resting membrane potential, if we remember, is minus 70 millivolts inside the cell, and essentially the outside is 0 millivolts because we're measuring it relative to one another. Okay, so yeah, this is super fun stuff we're talking about here, and if it gets confusing, just make sure you come to class with questions. Okay, so you're sitting at minus 70. What happens next? We've talked about resting membrane potential. What happens when you actually activate a neuron? Oh goodness. I'm talking for too long. So small graded potentials are essentially small deviations in resting membrane potential, meaning that they are what we call sub-threshold. So like you can see here, you've got your y-axis in millivolts, you have your x-axis in milliseconds, and a resting membrane potential at minus 70. And here you can see that it becomes slightly ever negative and then comes back. That is called a hyperpolarization. Hyperpolarization, when the cell becomes more negative. Okay, so hypopolarizing graded potential. You'll recognize here that the graded potential doesn't cause any action potential. It's just a slight dip. What could cause a graded potential? Well, there are two things. What would cause a cell to become more negative? You could have either a negative ion come in, something like chloride, or you can have positive ion leave, something like potassium. So if you have that positively charged potassium leaving the party, that's going to cause a hyperpolarization. Okay, so that's a hyperpolarization. Second, you have a depolarization. And depolarizations, again, you have that resting membrane potential of minus 70. You have the cell becoming more positive and then coming back down to your resting membrane potential. Okay, so this is a depolarization. What causes a depolarization? Well, you have the cell becoming more positive. So you could have essentially, this is in, this is in, this is out. Yeah, what could you have it going in the cell is potassium. I'm sorry, sodium. You could have sodium coming into the cell. You could have potassium coming into the cell. And like we saw with the previous slide, anions generally don't leave the cell. But I guess you could have chloride leaving the cell, but I don't think that happens. Okay, so these are graded potentials. We're talking about graded potentials because they are sub-threshold, meaning that they are not causing action potentials, which are really kind of like the interesting signal sending portion of this. Okay, graded potentials. So graded potential occurs in response to the opening of a mechanically gated or ligand-gated ion channel. So here we have the example of a mechanoreceptor. This is the mechanically gated channel. Pressure gets applied, and then you've got influx of ions. Again, that's probably most likely sodium or calcium. Next, you have, and this is a depolarizing. So if you were going to kind of measure the positive charge of this and actually kind of plot it, you would see it sitting at its resting graded potential, mechanically gated depolarization occurred, and you get a positive bump. It would be going up in the positive direction. So maybe from -70 to like, let's say -60, that little bump there. Okay, next we have a depolarizing graded potential caused by neurotransmitter acetylcholine. So you have acetylcholine binding, you've got that gate opening. And so again, you've got -70, it's sitting happily at -70. And then in the case of acetylcholine binding, it's a graded potential and just kind of, again, a little bump in the positive direction. It's becoming slightly more positive. Again, it's becoming more positive because you've got positive ions fluxing in. So you're going from negative to more positive. You've got positive ions flowing in, things like sodium. Okay, next we have a hyperpolarizing graded potential caused by a neurotransmitter glycine. So this is a different neurotransmitter. So what this neurotransmitter does is it allows for chloride to come in, okay? So you've got your little cell hanging out here at -70, and you've got more negative little ions coming in, and because more negative little ions are coming in, you're going to become more negative and return back up. So maybe you get down to like, let's say, -80. These changes are negligible. These numbers that I'm mentioning, you don't have to memorize the numbers. I'm giving you numbers so you get a sense of which direction the bumps are moving. I'm not going to say that a glycine receptor decreases or hyperpolarizes a cell to -80. Not necessarily true, but I just am giving you numbers so you can have a relative understanding of which way the bumps are moving, up or down. Okay, so fun stuff. I think it's fun. I'm sure you're just like, oh, is this fun? Okay, so the amplitude of a graded potential depends on the stimulus strength. So a stronger the stimulus is, the larger the bump in the positive or negative direction. So it just kind of depends. So when we talk about strength, it could be, in the case of a mechanical receptor, how much pressure is applied. In the case of those neurotransmitters binding, maybe the number of neurotransmitters that are binding. So again, you can graded. The reason they're graded, called graded potentials, is because again, the size of the stimulus correlates to the size of the bump. So stimulus 1, weakest stimulus, stimulus 3, the strongest stimulus, and that's causing the largest bump. Now we're making this contrast here because when we talk about action potentials, they're called all or nothing, meaning that once you've hit the threshold of an action potential, they're all going to look the same. So again, the graded potentials, stimulus strength, response to the size of the stimulus. Okay. So graded potentials can be added together to become larger in amplitude. So they're summative, or there's a summation. So if you have, let's say, a bump that is this big, and you add another bump that is this big, you can get a bump that is this big. And that's essentially what this is showing you here. Stimulus 1 is this big, and before it returns back down to the resting member potential, if you give a stimulus again, it's just going to kind of grow in size. So you can essentially give two stimuli close together and make that bump bigger. Or you can give a lot of stimuli at the same time at the first bump, and that can also become much larger. Okay, so again, don't get overwhelmed. Ask questions. We'll get to a place where this makes sense. Action potentials. So that's graded potentials. Now let's talk about action potentials. Action potentials. This is how I don't want to say majority of the signaling happens, but we're talking about neurons sending messages. Usually action potentials are the thing. So an action potential is a sequence of rapidly occurring events that decrease and eventually reverse the member potential depolarization and eventually restore it to resting state. So if we draw kind of a prototypical action potential, again, we're hanging out here. I'm just going to make this our minus 70 millivolts. That's where we're hanging out at our resting membrane potential. So let's say you're going along at minus 70, you get a graded potential, and then eventually at some point you reach what's called the threshold. So we'll say this threshold is minus 50. I think that's what we'll be using. Once you've reached minus 50, the cells become positive enough, it's reached threshold, and you have an action potential that fires. And it essentially comes back here to the resting membrane potential. So this here is considered your depolarization because you're becoming more positive. And then this is all considered your hyperpolarization because you're becoming more negative, and then you eventually come back here to your resting membrane potential. So we'll be talking a lot about this shape here in the subsequent slides. So hopefully some of you have seen this material before, and if not, that's okay. More than happy again to chat with you about it. Okay, so here's essentially the same figure we just went over. Okay, so how do action potentials actually happen? So you're hanging around at minus 70, you have a stimulus. That stimulus causes an influx of ions. So that stimulus is causing positive ions to come into the cell. And those positive ions hit the threshold. Now, at this point here, that threshold, why the threshold is important, because at that point, you have ion channels that are voltage gated. So again, if this was a cell membrane, and this is a channel, and it is voltage gated, meaning that once it hits minus 55, then you're going to have a ton of sodium fluxing into the cell. So once minus 55 hits, that gate swings open and you've got sodium coming in. That's what makes this far more positive here. So voltage gated sodium channels open and you've got this depolarizing phase. Now, somewhere around here, reversal of polarization minus, you have another type of voltage gated channel that opens up. And this is going to be the potassium gated voltage channel. This potassium gated voltage channel says, hey, it's getting way too positive in here. We need to kick some folks out. So at that point, that potassium is going to exit the cell. And as that positive potassium exits, it drives the cell more negative. So that's this repolarizing phase here. Now, that voltage gated potassium channel is kind of slow to close, like it doesn't just shut real quick. So because of that, you kind of have this little after hyperpolarization, meaning that the cell gets a little bit more negative than it would normally be, just because more positively charged potassiums have escaped during that time. Okay, so the key to this is resting membrane potential. Voltage gated sodium channels are in the resting state, meaning that they're closed. Voltage gated potassium channels are closed, and so that's this area here. Okay, and then green stimulus causes depolarization to threshold. That's this region here. And then this period that we see here is voltage gated sodium channels are open and the activation gates are open. So that goes. And then during this phase here, you have a voltage gated channels are open, sodium channels are inactivated. So these guys kind of essentially close, these guys are open. And so because of that, you've got the cell becoming more negative. And then here you have the voltage gated channels that are still open and the sodium channels are in the resting state. So again, you're talking about the activity of basically two different voltage gated channels. This voltage gated sodium channels open first, allow the cell to become far more positive because they're coming in. And then the potassium channels kind of are like too positive in here. Let's kick out some potassiums. And those potassiums hijack it and go outside of the cell. And so because of that, it makes the cell negative again and gets it back to that resting membrane potential of minus 70. Okay, so action potentials and stimulus strength. So action potentials can only occur if the cell reaches threshold. So like I said, here's our threshold. Here you have a graded potential. So you need enough graded potentials in order to reach threshold. At that point, you have an action potential. Once a cell reaches threshold, you have action potentials after action potential after action potential until it returns to resting membrane potential. Subthreshold stimulus, threshold stimulus, suprathreshold stimulus. Again, this particular figure right here, I do not think is an important point for you to know. We can talk about this if you like, but it's kind of more specific than we need to be at this point. Okay. So in terms of kind of breaking down those voltage gated channels. Okay, so at resting state, you have your voltage gated sodium channel, voltage gated potassium channel. Both of them should be closed. Depolarizing stimulus happens. Voltage gated potassium open or sodium opens and sodium comes inside the cell. So sodium's coming inside the cell, making it far more positive. Now, at this point, you've reached kind of pretty high up on the peak. The repolarizing phase begins. So your voltage gated sodium channels close, your voltage gated potassium channels open, and you've got positive charge leaving the cell, making the cell more negative. So at this point, you're going to start to see that turn in the peak. And then lastly, during the repolarization phase, you've got continued leaving of the potassium out the voltage gated potassium channel, and then eventually you will see the voltage gated sodium channels become players in this again. Okay, so comparisons of graded and action potentials. Okay, so duration. So graded potentials are longer. They can be several milliseconds, and then action potentials are very short. Polarity, maybe hyperpolarizing, meaning more negative, or depolarizing, more positive. Action potentials are always going to be more positive because they're mediated by that change in the positive direction. And the refractory period, that doesn't really occur for graded potentials because there's no ion channel that's slowly closing because you're not really competing with two different ion channels. And then with action potentials, you do have that refractory period, or that period in which the potassium is able to still come into the cell. Okay, so we talk about the neuron sending signals. We can see that there is continuous via and saltatory conduction. So what does that mean? It means that as an action potential is traveling down an axon, there are two different ways in which that it can occur. It can occur in a continuous way, meaning that you just have voltage gated sodium channels that are open as the depolarization moves down the cell. Or you can have saltatory conduction, which in the case of myelinated, this is how this is different, because these are myelin nodes of Rabier. These are chunks of myelin. Again, this makes it far more easy to conduct this message. So again, at that point in time, you've got the action potential moving through this, jumping this kind of nodes of Rabier, and then you have it jumping again. Again, this is maybe a level of specificity that we do not need to hit for this course. So I'm not going to ask you about continuous versus saltatory conduction, but I'm more than happy to chat about it with you. I think it's just kind of outside the scope. Just important takeaway from this slide, there are myelinated and unmyelinated axons. Myelination increases the speed of conductivity. Okay, so things that affect propagation speed. So factors that affect propagation speed, things like exon diameter, amount of myelination, and temperature. So when we talk about propagation speed, we're basically talking about, okay, if you have a action potential traveling down an axon, how fast does that happen? Does it happen kind of slow, or is it basically going not at the speed of light, but pretty darn fast? Think about exon diameter, kind of the smaller it is, the kind of the faster it's going to go, and the amount of myelination. So if you have a piece of axon that's completely myelinated, that's going to increase the speed of conductivity and temperature. So the warmer something is, the faster it's going to conduct as well, because just kinetic energy increases propagation speed. But again, probably not going to ask you a ton about this. Myelination and conduction rates, if you're interested in kind of the comparing and contrasting about myelination, by all means. Okay, signal transmission at synapses. You're only halfway done. So when we are talking about action potentials and sending signals, so like we talked about in the first lecture, there are these cell bodies with these dendrites all hanging out, and they're getting inputs from different locations. And then there's that one axonal projection that sends the action potential down like that. Now, once it reaches the end here, this has to be turned from an electrical signal into a chemical signal. So again, it's going to be reaching out and sending signals onto the dendrites of the next cell. And then again, kind of continually does the same thing. And you've got your axon So this is chemical, and then it turned, oh gosh, this is not great drawing. Chemical, and then again sends it. And so this area here is called the synapse. We're going to talk about this area now. My drawing here is not the best, so we're going to just keep moving forward, and the figures are going to probably be far better than the crazy drawing I just put together. Okay, so there are two different types of synapses. There are electrical synapses and there are chemical synapses. Now, electrical synapses we probably won't talk about a ton. They do occur in the body. They're important for sending very rapid signals. If you go back and look at the lecture on junctions, gap junctions are actually a type of electrical synapse. And so you literally have these two cells that are just basically sandwiched right next to one another. And you've got these ion channels that are opening and closing within that sandwich. And you've got ions fluxing straight into that next cell. So it's a very rapid way to for cells to communicate. It's important for synchronizing activity. So your heart cells are a great example of a place that has electrical synapses, because it's important that that actually happens in a very synchronized way, your heart, your muscle contraction. Chemical synapses are the type of synapse I was just describing, where you have an electrical signal that gets turned into a chemical signal and then back into an electrical signal. Terms on this slide that maybe you don't know, things that are something like a presynaptic and a postsynaptic neuron. Okay, so I'm going to draw this again. I'm sure the next slide has the perfect figure on it. So you've got a cell body, dendrites, the axonal projection, and then you've got kind of the end of the axon where the chemical signal is going to be released. Now, this is called the presynaptic neuron. Presynaptic because it precedes the synapse. The synapse is essentially the distance between one neuron and another neuron. So the synapse is that kind of little area between the two here that the neurotransmitters have to travel across. So that's the presynaptic neuron. We'll call this the synapse because it is. And then this cell that is receiving the information is called the postsynaptic neuron. So presynaptic neurons segment the signal travels across the synapse, the postsynaptic neuron receives the signal. Okay. Again, figures here, probably a little bit better than what I was just drawing. Okay, so when we talk about kind of the interaction between synapses, we often think of axonodendritic, meaning that there's an axon. This is an axon of a cell up here that's sending its signal down. It's projecting onto the dendrites of this cell. So axodendritic, meaning axon and then dendrite. You can have an axon-axonic, meaning that you have a cell that's actually projecting onto the axon itself. And then you can have an axosomatic, meaning that you've got a cell sending a signal and actually releasing neurotransmitter onto the body of the cell. Now, normally what we're going to be looking at is axodendritic. It's probably just the most prototypical type of interaction that happens. And so for the remaining, I think probably the remaining slides we'll be focusing on axodendritic projections or interactions. Okay, events of the synapse, again, very dynamic process. Go ahead and watch the video. Okay, so if we go ahead and zoom into the synapse and check out what's happening there. Here you can see that you have a action potential traveling down that axon. So axon. So that potential travels down that axon. Again, it's making the cell more positive as it does that. So as it travels down to the end of the axon terminal, or the synaptic end bulb, this is becoming far more positive. And we've got, oh my gosh, voltage-gated calcium channels. So once this little area becomes far more positive, these voltage-gated calcium channels open, and you've got calcium fluxing into the cell. So calcium fluxes into the cell. And once that happens, you have these synaptic vesicles, but are just basically little pockets of neurotransmitter that end up essentially fusing or kind of just merging onto the end of the cell and releasing its little neurotransmitters out here into the synapse that travels across the synapse, binds to the postsynaptic receptor, again, the receptor or the cell that's receiving the message. And so at that point in time, you've got if these are ligand-gated ion channels. That neurotransmitter is going to bind to that receptor. And once it happens, you've got ions fluxing into the cell. So at that point in time, you're passing that, electrical signal into a chemical signal. And at that point in time, it becomes electrical again because you've got ions fluxing into the postsynaptic cell. Oh, goodness. Again, this is kind of a long lecture with a lot of material in it, so hopefully it does not become overwhelming. And if you're feeling overwhelmed, pause the lecture, come back to it later. That's a great thing about this being on YouTube. Okay, so quick recap. Action potential travels down, makes the cell more positive, causes voltage-gated calcium channels to open. That causes release of neurotransmitter from the presynaptic cell. The neurotransmitter travels across the synaptic cleft, binds to the ligand-gated ion channels on the postsynaptic cell. That causes those channels to open. It causes influx of ions, causing a depolarization or a more positive kind of influx of the charge on the postsynaptic side. And so that's, again, how this whole process starts all over again, is you've got those positive charges fluxing in. Okay, so as the cell becomes more positive, as if we didn't have enough things to kind of remember. Okay, remember when we talked about things, degraded potentials, depolarizing, becoming more positive? Well, these are also called excitatory postsynaptic potentials. It's a depolarizing postsynaptic potential. So if you were looking at the postsynaptic cell as those positive ions flux in, this is an excitatory postsynaptic potential. Again, because you have positive ions fluxing in. Now, let's say you have an inhibitory neurotransmitter that's binding. Those inhibitory neurotransmitters cause negative ions to flux in, so that causes a hyperpolarizing graded potential. So this is depolarizing, this is hyperpolarizing. So hyperpolarization happens in response to an inhibitory neurotransmitter. So that's called an inhibitory postsynaptic potential. Excitatory neurotransmitters cause depolarizations. Inhibitory neurotransmitters cause hyperpolarizations. And that's because you have negative ions, such as, or most often, chloride. And in the case of excitatory neurons, most often that's sodium coming into the cell. So most often it's excitatory because it's exciting the cell, it's making it, more likely to fire an action potential. That's influx of sodium. And in the case of inhibitory neurotransmitters, you have influx of chloride, making the cell more negative and making it essentially less likely that an action potential will fire because the cell is now more negative. Okay. A postsynaptic neuron can receive many signals at once, meaning that it's got all those dendritic projections, and sometimes it's getting positive inputs, sometimes it's getting negative inputs. It can be receiving both excitatory and inhibitory postsynaptic potentials at exactly the same time. So it's almost like you're living out there in the world, right? And you've got friends who are super exciting and just not exciting, super positive and just like, yay, world. And then you've got friends out in the world that are just kind of like, you're just not super exciting. So if you're kind of the cell body, it's just like you take in all of these inputs and then that it's going to affect kind of your overall affect. So sometimes you're going to have a lot of positive inputs from your happy friends, and sometimes you're going to have a little bit more ER inputs, which might also make you sad. Again, maybe that you find that analogy helpful, maybe not. Okay, so now we're making our way into talking about neurotransmitter receptors. Okay, so neurotransmitters at chemical synapses can either be excitatory or inhibitory graded potentials. So we were just talking about excitatory and inhibitory neurotransmitters. Like I said, excitatory neurotransmitters cause generally influx of sodium. Inhibitory neurotransmitters generally cause influx of chloride. And you can have two different kinds of neurotransmitter receptors. You can have ionotropic receptors. Okay, what does this word look like? Let's start there. Ionotropic looks like the word ion. So this is where you have influx of positive or negative ions, cations or anions. And then you have metabotropic receptors. Metabotropic receptors are kind of considered the slow receptors. Now these receptors, I'm going to draw them in this way, but this is the receptor, this is the plasma membrane, and they basically bind neurotransmitters up here on the outside. And then they got these little like messenger systems attached to them called G proteins. We'll talk about this more, I'm guessing. These G proteins, and those are going to go off into the cell and send signals. They're basically going to activate signals in the cell. So this is kind of like much more signaling cascadey. And this up here is far more just ions go in and a change happens. Ions go out and a change happens. So this is ionotropic receptors are generally considered the fast receptors, and then metabotropic are the slow ones. And so as if this weren't confusing enough, a new single neurotransmitter can have both of these types. So for example, let's say a neurotransmitter called glutamate can bind to both ionotropic and metabotropic receptors. We'll talk about that more in a bit. We'll just focus on the fact that ionotropic are fast and have ions that flow through, and that metabotropic are slow and cause activation of these receptors that have these kind of little minions that go out and send signals inside the cell. And that just takes a lot longer than ions moving in, but still does cause an effect to the cell. Okay, so ionotropic and metabotropic receptors. So here we have an example of the acetylcholine receptor. This is a ligand-gated ionotropic acetylcholine receptor where acetylcholine binds to that receptor, opens it up, and you've got movement of ions inside and outside the cell. This is causing an excitatory postsynaptic potential because you've got positive ions going into the cell. Okay. Next, we have the ionotropic GABA receptor. So GABA is an inhibitory receptor, meaning it makes the cell more negative. So once the neurotransmitter binds, the channel opens, and then you've got influx of negative ions. So you've got the inside of the cell becoming more negative. So if we are going to draw these on our XY axis, you've got your resting membrane potential. This is becoming more positive. You've got your resting membrane potential. You're becoming more negative. This is an excitatory postsynaptic potential that is depolarizing. This is an inhibitory postsynaptic potential that is hyperpolarizing. We're not just doing enough new vernacular. Good times. Okay, lastly, we have a metabotropic acetylcholine receptor. So the metabotropic acetylcholine receptor has a ligand that binds. And so like I said before, there are, a single type of neurotransmitter can have both ionotropic and metabotropic. So metabotropic receptor binds. This little guy hanging out down here, this G protein, gets released and goes and does its thing, sends signals, and can cause effects on things like the potassium channel. But again, this whole process here is very simplified, but can cause voltage changes in the cell based on its interaction with other ion channels. Okay, clear as mud? Awesome. You better come to class on Thursday, man. We're going to talk about this a ton. Super fun time. Okay, so removal of a neurotransmitter. So you send this neurotransmitter out into the synapse. How do you stop it from signaling? Okay, there are a few different ways that you can do that, right? First way is through diffusion. So that neurotransmitter we've got our presynaptic cell, we have our postsynaptic cell. Our presynaptic cell releases neurotransmitter out into the cell, out into the synapse. Okay, so how do we get rid of that? Well, it can just diffuse away outside of the synapse, away from it, so it no longer hits this postsynaptic cell. Second way is enzymatic degradation, meaning that there is outside of that cell a little Pac-Man enzyme that basically eats up the neurotransmitter and breaks it down so it's no longer signaling. It's kind of cool. So it breaks down that neurotransmitter and gets rid of it. So no more neurotransmitter, little Pac-Man enzymes have broken it down. Last way is uptake into cells. So if we remember when we were talking about different cell types, these are going to be our astrocytes, glial cells. Remember those? So as we have neurotransmitter being released out into the cell, these little glial cells here have essentially little vacuum cleaners on them that are sucking up that neurotransmitter into the inside of the cell. So that neurotransmitter gets sucked up into those cells and can also get sucked up by the neurons as well. But for the most part, gets sucked up by the glial cells. So that's one of the ways that the glial cells support neurotransmission is by sucking up that excess neurotransmitter into them and essentially stopping the signal from being sent. Okay. Summation. So if several presynaptic bulbs release their neurotransmitter about the same time, the effect can be generate a nerve impulse due to summation. Summation can be spatial or temporal. Okay, I'm gonna draw this neuron again. It's got dendritic projections. Okay, so in the case of summation, summation is kind of exactly what it sounds like, is you're adding things together. So you can either, what is called spatial summation, you can essentially cause activation of dendrites simultaneously, all happening at once. So if all of these dendrites receive a bunch of positive depolarizations all at the same time, that's going to cause this cell to send an action potential. Now, that's one way that this can work. That's called spatial summation because they're all happening in different places, but they're all, again, summing all together. So it's almost like if you were a cell and you kept on hearing a rumor from a bunch of different people, but all at the same, but a bunch of different people, but kind of at the same time. So you've got the rumor coming from this person, this person, this person, this person, and this person. So at that point, you're just like, I got to pass this rumor on. Boom, you fire an action potential. Okay, so that's spatial summation. The second type of summation is temporal summation. So temporal summation is different in the fact that you don't have it occurring at a bunch of different dendrites at the same time. In the case of temporal summation, you just have it occurring a bunch of times at a single site. So here you have boom, 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 a bunch of summations happening at the exact same time. And that causes you, person, to fire an action potential. It's almost like if you have that one unrelenting friend who keeps telling you the same rumor again and again and again and again and again, and finally you're like, fine, I will pass this on. So again, this is kind of the unrelenting firing of a single neuron. And so that's temporal summation, because it's called temporal because it all occurs at a very specific point in time, temporally time, okay? Okay, spatial summation, again, it's showing you here a little bit of the drawing that I just did, where you've got, this is your first axonal dendritic projection, this is your second. And so if you have, activation here, then activation here, and then many, you can have an AP that actually fires at two different locations because you're summating from two different places. Here, you just have a single presynaptic neuron synapsing onto this single postsynaptic neuron. And again, you just have repeated stimulations. That one unrelenting friend who's like, da 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 da, you have to pass this on. Here's what that looks like. Okay, summation, summary of neuronal structure. Dendrites, cell body, junction of the axon hillock. So dendrites project out into the world, have channels and ligand-gated and mechanogated channels on them. They're basically reaching out into the world looking for a stimulus. Next, you have cell body, which receives stimuli and produces excitatory postsynaptic potentials and inhibitory postsynaptic potentials. And then lastly, here you have an axon hillock, which is going to be the region of the cell which decides to essentially go or no-go that. But we haven't talked about the axon hillock. Maybe we'll cover that in class. Okay. The axon is going to be this projection down here. Axon terminals is down here. These are all of your dendrites. So, and if you're wondering kind of more about that axon hillock region, that's kind of located right here at the base of the axon. It basically is the go or no-go place of whether you're going to send an action potential or not. So here you have plasma includes chemically gated channels. Again, you have you're reaching out to have neurotransmitters bind to those dendrites. Here you have your voltage-gated sodium and potassium channels because that's what mediates action potentials. Then at the very end of these bulbs down here, Those are voltage-gated calcium channels, again, because that's where you're going to be releasing neurotransmitter from. Feel like neuroscientists yet? Okay, neurotransmitters. So we keep talking about these neurotransmitters that diffuse across the synapse. Okay, how do we characterize those? Okay, so small molecule neurotransmitters. We have acetylcholine, which essentially is in both your central and peripheral nervous system. It can be, it can have both metabotropic and ionotropic receptors. Amino acids, these are very important. Majority of the signaling in your brain is mediated via these amino acids. Those amino acids are things like glutamate, GABA, and the other one is going to be glycine. And these are very important. Again, these mediate a majority of the synaptic transmission. Everything else is kind of neuromodulatory, meaning that that just kind of tunes the signal a little bit. Biogenic amines, ATP and other purines, nitric oxide, and carbon monoxide. We're going to have to pare this down and make this far simpler for you guys to memorize. We'll talk about the important neurotransmitters and highlight those. Okay, so here's some of the structures. This is not a chemistry class, so you do not need to memorize these structures. Acetylcholine is the first one that we talked about there. There are receptors that bind nitric oxide, carbon monoxide, purines such as ATP, neuropeptides. Again, this is just a protein. You can tell that because these are little amino acids here. The amino acids, like I said before, glutamate, aspartate, GABA, glycine. Glutamate is the major excitatory neurotransmitter. So pretty much every thought you're having in your brain right now is being mediated by via glutamate. It's the major excitatory neurotransmitter. That is something you'll need to know. GABA is the major inhibitory neurotransmitter. So if GABA was the brakes, glutamate would be the gas. Glycine is also a neurotransmitter. It is also a inhibitory neurotransmitter. It puts on the brakes, but probably not as much in the central nervous system as GABA. Okay, neuropeptides. This is just kind of an FYI. You will not need to know anything on this slide. Substance P, enkephalins, endorphins, dynorphins, hypothalamic releasing, angiotensin 2, cyclocystokinin. These are neuropeptides. Again, don't need to know for this particular chapter. Neuropeptides, again, it's kind of giving you a description and some of the effects of those neuropeptides. You can see that there's a huge relation to pain. These peptides are binding receptors to mediate pain. Again, this might be more in the peripheral nervous system because we actually don't have pain receptors in our central nervous system. So mediated in the peripheral nervous system. Neuropeptides, angiotensins, CCK, neuropeptide Y. Again, neuropeptides, I'm not going to go over that a ton. Okay, neural circuits. Okay. Sorry, just trying to figure out how to pace out this last part. Okay, neural circuits. So you can have, the way that the circuitry in a brain is kind of put together can have a variety of structures. You can have a simple circuit, a diverging, a converging, or reverberating parallel after discharge. So in terms of diverging and converging, you can have in the instance, usually from signals being sent from your central nervous system into your peripheral, a diverging kind of pattern where you have a single neuron that's releasing input into more and more and more outputs, versus a converging circuit where you have multiple inputs onto a single neuron. And again, just kind of depending on the placement of these neurons is going to, structure is going to essentially affect function. Other types of circuitry, reverberating circuits, where you have circuits that are kind of looping back onto themselves. This is very much a regulation type of circuit. We see this probably a little bit more in like the peripheral nervous system and the spinal cord. And then here you can see the parallel after discharge circuit. You've got a single neuron that's sending a signal out, and then you have it kind of synapsing onto kind of signal neurons to its left and its right. And it's like, okay, let's make sure this neuron gets a signal. It's going to all converge on that output at the same time. Might be a great example of spatial summation. We just talked about that. Okay, regeneration, repair of nervous tissue. So the brain is an interesting thing because for the most part, neurons are not super, let's say, hardy in terms of regenerating themselves for the most part. There are exceptions to that, especially in the central nervous system. The peripheral nervous system is slightly different. But what's cool about neurons is that they're constantly remodeling themselves and creating new pathways and new connections. And so we call that remodeling and the changing and the forming and new connections plasticity. And so plasticity is actually kind of the molecular mechanism of how we learn. Right now, as you're learning this, you are going to be forming new neural connections within your brain, and that's your brain's plasticity. Plasticity is much greater the younger you are. It is a known fact. That's why kids can learn things so quickly, is because our brains are super plastic. They're constantly forming and unforming these neural pathways. And then in terms of regenerating, it's a capability to replicate or repair. And again, not super frequent happening in the central nervous system. There are some exceptions to that rule, but for the most part, not a super ton of regenerative things happening in the central nervous system, but the peripheral nervous system is a little different. You do see a little bit more regeneration there. Okay, neurogenesis, or the generation of new neurons, basically doesn't really happen very often. Inhibitory influences from neuroglia, especially oligodendrocytes, stops that repair from happening. Absent of gross stimulating cues that were present during fetal development. Again, that's what causes these neurons to divide. And so that's what's happening and upregulated during fetal development, is you've got a ton of brain neurogenesis exploding. And then you also have rapid formation of scar tissue. And so ultimately, neurogenesis occurs in those much younger animals, people, and not so much in older ones, but there are a couple different regions in your brain neurogenesis does continue to happen. And I will tell you, there are some activities that can actually increase neurogenesis, one of them being exercise. So just put that in your brain bank and remember that. If you want to stimulate new neurons in your brain, exercise is a great way to do that. Okay, damage and repair in the CNS. So the peripheral nervous system, like I said, able to repair itself pretty easily. But in terms of the repair process, we've got chromolysis, wall degeneration, and formation of the regeneration tube. We are not really going to talk about damage and repair too much. So again, gonna just pass on by this. Okay, disorders. This is such an abbreviated list of disorders for the nervous system. Multiple sclerosis, again, one type of disorder that affects, that could be a result of demyelination. Again, if you lose that myelin, the speed in which neurotransmission occurs is very, can be abbreviated, can be very short. Epilepsy is basically a kind of a blanket type of diagnosis for seizures. So seizures can be generated for a whole variety of reasons, varying in terms of whether they're just intrinsic to the individual or they're brought on by a head injury or things like that. So epilepsy, again, can be caused by a variety of things. And that's misfiring of the brain. You just have a bunch of, it's like a short circuit happens and then it just, everything kind of boom, 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 boom happens. Excitotoxicity, that's when parts of your brain undergo damage and it releases a ton of neurotransmitter that shouldn't be there, and that causes, again, kind of a misfiring and miswiring in the system. And then depression, again, this is such an abbreviated list of disorders. Depression would be caused by kind of misregulation of certain neurotransmitters, so things like serotonin and dopamine that regulate emotion can sometimes be the result of, can be the cause of depression. But again, depression is a very blanket diagnosis and can be caused by a whole variety of things. Like we're talking about epilepsy and multiple sclerosis. Those could even be, you know, other diseases that occur with depression as well. Post-traumatic stress disorder could be the, you know, a cause for depression. You know, there again, there's are such complicated disorders because you've got so many different factors that play into the wiring of the brain. Okay, I think we might be, we're done. That was a long one. Again, if you feel the need to stop and pause and come back to this lecture, please feel free to do so. And we're going to be talking about this all week. And I am super excited about this topic and I love it, but I can even feel myself starting to like, this is long at the end of the lecture. So if you have any questions or concerns, feel free to reach out. I'm super happy to answer and clarify anything that we went over today in lecture. And I hope everyone has a great day.